The neuromuscular junction consists of a nerve end, a muscle end plate, and a space in between known as synaptic cleft. The neurotransmitter at neuromuscular junction is acetylcholine. Acetylcholine is stored in the vesicles of the nerve. When there is stimulation of the nerve, these vesicles migrate to the surface, ruptured, and then releases the stored acetylcholines. Now the acetylcholine molecules bind to a receptors on the muscle end plate known as acetylcholine receptors, which leads to opening of ion channels so that ions can move across the membrane and depolarize it, producing end plate potential and ultimately triggering muscle contraction. Each neuromuscular junction contains approximately 5 million of these receptors, that is, the acetylcholine receptors, but activation of only about 500,000 receptors is required for normal muscle contraction. Now, understanding of these acetylcholine receptors is quite crucial in understanding how the acetylcholine works, as well as how the neuromuscular blocking agents works. The structure of acetylcholine receptors varies in different tissues and at different times in development. Each acetylcholine receptor in the neuromuscular junction normally consists of 5 protein subunits, 2 alpha subunits, and single beta, delta, and epsilon subunits. Only the 2 identical alpha subunits are capable of binding ACH molecules. If both binding sites are occupied by acetylcholine, a conformational change in the subunit opens an ion channel in the core of the receptor. In contrast to the normal junctional acetylcholine receptor, another isoform contains a gamma subunit instead of the epsilon subunit. This isoform is referred to as the fetal or immature receptor because it is in the form initially expressed in fetal muscle. It is also often referred to as extrajunctional because, unlike the mature isoform, it may be located anywhere in the muscle membrane, inside or outside the neuromuscular junction when expressed in adults. Now, when sufficient receptors are occupied by acetylcholine, the end plate potential will be strong enough to depolarize the perijunctional membrane, that is, the areas around this neuromuscular junction. Voltage-gated sodium channels within this portion of the muscle membrane open when a threshold voltage is developed across them. The resulting action potential propagates along the muscle membrane and T-tubule system, opening sodium channels and releasing calcium from the sarcoplasmic reticulum. This intracellular calcium allows the contractile proteins actin and myosin to interact, bringing about muscle contraction. The amount of acetylcholine released and the number of receptors subsequently activated with efferent nerve depolarization will normally far exceed the minimum required for the initiation of an action potential in the muscle. The nearly tenfold margin of safety is reduced in Eaton-Lambert myasthenic syndrome, in which there is decreased release of acetylcholine, and myasthenia gravis, in which there is decreased number of acetylcholine receptors. Now, the sodium channel is a transmembrane protein that can be conceptualized as having two gates. Sodium ions pass only when both gates are open. Opening of the gate is time dependent and voltage dependent. The channel therefore possesses three functional states. At rest, the lower gate is open, but the upper gate is closed. When the muscle membrane reaches threshold voltage depolarization, the upper gate opens and sodium can pass. Shortly after the upper gate opens, the time-dependent lower gate closes. When the membrane repolarizes to its resting voltage, the upper gate closes and the lower gate opens. The neurotransmitter acetylcholine is rapidly hydrolyzed into acetate and choline by the substrate specific acetylcholinesterase. This enzyme is embedded into the motor end plate membrane immediately adjacent to the acetylcholine receptors. After unbinding acetylcholine, the receptor's ion channels close, permitting the end plate to repolarize. Calcium is resequestered in the sarcoplasmic reticulum and the muscle cell relaxes again.